Great. Okay. So we'll get started. So just a reminder, probably everybody's been to a demo by now, but welcome to this package demo. We're going to have Jiaji Chen, Matthew O'Brien, and Joselyn Chavez talk about this Giotto Suite. And if you have questions for the speaker, please enter them in the Q&A tab on the platform. I will be monitoring that during the demo, and I'll just bring those up. And if you're in the room, you can just raise your hand. The demos are meant to be interactive, so please feel free to ask questions throughout. Okay, go ahead.
Thank you so much. Hi, everyone. My name is Jiaji Chen, and I'm the lead developer on Giotto Suite. I'm here to talk a little bit about our platform today, which is a technology-agnostic framework for spatial omics data analysis in R. I'm extremely thankful for the opportunity to present here at BioConnector 2023. With me today are two other members of the core development team for Giotto, and that's Matthew O'Brien, who's the bioinformatician of the Dries lab, and also Joselyn Chavez from the Yuan lab, our collaborators at Mount Sinai, who's a postdoc. And now I'll just hand it over to Matt, who's going to talk about and give an overview of Giotto and also the rest of this presentation.
All right, everybody, before we get started, I'd just like to orient us to the rest of this presentation. So we're going to start out with some background. George is going to take us through the structure of the Giotto object and run us through a mini data set from the VisGen MERScope doing a demo. I'll take us through the Zenium breast cancer data set demo. Joselyn will show off some of the multimodal integration and interactive selection capabilities. George will again speak a little bit to the interoperability efforts that we've put into this project, and then we'll wrap up the packet, or we'll wrap up with some conclusion statements. Just so that everybody is aware, the scripts and demo files are available on GitHub right now. It's the Dries lab /Giotto_BioC_2023. So over the past couple of decades, spatial technologies have been emerging with not only increasing capability, but also increasing rapidity and increasing throughput. I apologize. I'm going to make this. Can we do this? I believe, yeah, everything is still visible. Yep. Wonderful. So these technologies encompass multiple different outputs and have multiple different experimental strategies in order to generate data. Some methods of note include seqfish, merfish, nanostrings, cosmics, 10x's Zenium, and extremely notably for its accessibility and how widespread it is throughout the field right now, 10x's Visium platform. And so going hand in hand with the emerging technologies over the past couple of years, spatial data generation has also accelerated greatly in recent years. Ruben Dries, the leader of the Dries lab and the creator of Giotto Suite, was in 2018 developing some scripts to handle some of the spatial data for a specific technology when he realized that this was applicable across all spatial technologies, which led to the creation of Giotto. So Giotto was created, as I just said, to handle spatial data of all types, multiple of which are listed on the screen right now. This was detailed in the initial release and in a previous manuscript. So representing this kind of data as opposed to single-cell data, single-cell RNA-seq data, it comes with its differences. Namely, we have now spatial coordinates in X and Y. So we can represent this data in the form of grids, or alternatively, we can represent it using networks based on k neighbors and or distance, and then formulate a network using Delaunay triangulation. Giotto streamlines the analysis of spatial data in methods such as spot-level deconvolution, cell type identification, cell-cell interaction, or cell ligand interaction, and multiple others, which we detailed in a previous manuscript as well. However, challenges remained when representing the spatial data, namely that biology does happen at multiple scales and across multiple tissues, which all function in 3 dimensions. Furthermore, each of these technologies has its own format, throughput, and resolution, which leads to multiple different data types to represent exactly what is going on within the biology itself. And finally, the cellular and tissue phenotypes are determined in a multimodal manner. It is more than just the transcriptomic information that is going to explain the phenotype. And so that leads us to the question, how can we build a unified and standard data structure to represent all technologies and their respective data efficiently? To answer this question, we would propose Giotto Suite, the framework of which George is going to take us through right now.
So Giotto Suite introduces two concepts that largely define like how the entire object is put together. And these, I'm going to start with spatial unit for this. So the spatial unit is essentially like what your actual unit of analysis is. So for previous technologies such as, or for certain technologies such as single cell analysis or for Zenium, you're kind of already locked into kind of a mutable unit of study, right? So you're locked to either that single cell or you're locked to just that Zenium spot. But with some of the newer technologies that are coming out with multiplexed immunofluorescence or like multiplexed imaging, you actually are free to pick specifically which sets of annotations you want to use. So you could say, for example, you only care about what's going on in the nucleus, or you care about the entire cell itself, or even like morphological regions that are larger than that. So this opens up a lot of flexibility for how you want to organize or also analyze your data. And the other aspect that we're introducing for this is already something that really exists, but it's the feature type. So this is basically what modality information you're looking at. So it can be protein information, it can be RNA information. And these over here are just some examples of very raw types of this kind of data. For example, you have point data for RNA detections, you have intensity data for protein information, and you can also have integrated modalities. So that's also something that we would call a feature type. So all of this is like our framework for organizing all the different information that goes into the Giotto object that we append into it, and also how they can potentially be put together even. So this is that latter step where you have your feature types and also you have your spatial units, and now you want to put together an aggregate analysis. So a better representation of how that could work is like if you have feature points and you have intensity data over here, and you also have a set of polygon annotations, whatever these might be, you would then aggregate them based on points for counts data, or you could aggregate that based on mean intensity in order to finally get your aggregate data, which is going to be your expression matrix. So this one I'm just going to touch on really quickly, but I'm going to explain later in more depth during the demo. So this is just an additional thing that we track for when we want to combine multiple spatial units together. So this is going to be in cases where we might have, for example, multiple Z layers. And then we want to basically collapse all that information together into a single final data set that we're actually taking a look at. So I'm going to move on to the demo right now, and this is going to be over here, and I hope this is still visible. Okay. So this is kind of like the overall design language of the Giotto object, and also it's going to walk through how you would put together an analysis from the very beginning for subcellular data sets. A lot of this is just an explanation of the philosophy of the Giotto object, and also the spatial unit feature type that I already talked through. And this over here is an example of basically the internal organization that we have within the object. So you have the Giotto object here, you have a slot here, and that could be, for example, your expression information, and then that would further be subnested by your spatial unit, and then the feature type. So the feature type like owns whichever modality additional analyses that are falling under it. For example, like you could be looking at your cell nucleus over here, you could look at your entire cell here, and then underneath that unit that you're actually studying, you would have additional modalities for whichever one you're actually taking a look at. Yes.
The font size.
Oh, is that better?
Yeah.
Okay. Great. So this is just the setup, really. This is how you would install. A lot of these scripts are going to have this at the beginning. And then, so the first step I'm going to talk about is actually creating the Giotto object. So this is going to be loading the information. And the information that is needed for a Giotto object analysis is going to be a set of expression information and spatial information. For stuff like Zenium, that's going to be something like your expression matrix plus paired spatial locations. And for something like subcellular information, it's going to be a set of raw feature data like feature detections and images and polygonal information. And this particular tutorial is going to talk through how you would put together a subset of the vision merscope mouse brain receptor map. So that's going to data release that we've already subset down in so that it's easy to load in and play around with. And this is available as part of our additional package called Giotto Data. So it's handy for anybody who wants to take a look at that. Okay. So also to basically represent these new types of information for subcellular information, we are leaning pretty heavily on the geospatial field. They have a lot of existing objects that already are very good representations for this kind of information. So mostly spatial vectors, spat vectors from Terra, and also spat rasters from Terra. And the Giotto polygon and Giotto points are essentially extensions of the spat vector that Terra has. So this is our representation of the polygonal information for cells, and this is our representation for the points, the transcripts that come out of it. And since a lot of this already builds on pre-existing framework, we are also able to port over a lot of the functionality for things such as like spatial manipulation, flipping, XY translation, rotation, even for spat vectors specifically anyways. And then we also have the images themselves, which are very efficient in terms of how much memory it takes in the session itself, and also it plots very quickly because it only like grabs a certain amount of pixels in order to, that are needed for you to actually make the plot itself. So jumping into the object creation itself, the subcellular workflow is usually after you create your object using create Giotto object, you attach your feature information, your spatial information, and then after that you would add in your spatial locations as basically a set, as the centroids of those polygons that you originally had. And following that, you would attach in your images. So this is just a plot showing you that you have your images and they're well aligned with the polygonal information that you do have. And then following that, similar to what I showed earlier with that figure that where you are able to aggregate all of your points data using the polygons, basically count up all the ones that fall within. We perform the step of aggregating to get our expression matrix. So this is going to be done with the Z0 set of polygonal information using all the RNA information. And this is what that looks like. So we've turned those point data into a count matrix, essentially. And you can further explore this object now that we've made it. So you can directly returning it will give you a summary of what is actually attached to the Giotto objects. You have your expression information created, and your spatial locations, and your images. And taking a closer look at it, you can use the show functions that we have built out for most of the slots. I think all of them at this point. And that's, you have the Z0, you have RNA, and then this is your raw values matrix for your expression information. So that's a preview of what that looks like. So I've also been misleading you a little bit so far about like how you would want to treat this particular data set, because this one's a little bit special in terms of the information that it contains, because it has multiple Z layers to it, right? So you have, actually for this particular data set, you have 7 different layers of polygonal information and also of transcript information. So how do you really deal with that, right? Well, in Giotto, we treat it as essentially like multiple layers of aggregation you would do, and then after that, you would collapse it down. So the first step is to remake your Giotto object over here. I'm just remaking it because to show like we've done it wrong the first time around, because we're essentially looking at it as a cylinder as opposed to like a volumetric cell, right? So we would redo the aggregation as a set of like Z0 and then Z1, just for the sake of time. We would not go through all 7. And then additionally, these are params that we did not use last time during aggregation. And this is specifically used to subset a set of metadata that was attached to the points information. So I neglected to mention that earlier, but the points information actually for each point has a set of metadata that can be attached to it as additional values. And then you would collapse this all into your a common aggregate spatial unit. So what that means is that you've made all of these aggregations. You want to just sum all those values together. And then after that, you would also get the mean locations of the vertices and produce a final set of polygons to represent your information. So now you've got like a better volumetric information produced. This is just more printouts for what we've just made. Oh, and also, because we're leaning heavily on the spatial field for these sets of analyses, we can also tap into some of the analyses that they already have. So one of which is spatial autocorrelation. And this is, if you have genes, you can correlate them. But on top of that, this is spatial information, right? So you actually have to care about how close they are to each other so that correlation actually matters, right? So that's what spatial autocorrelation is. And we have global statistics and also local statistics that we can run. So this is just an example of how you would run Moran's eye for that set of data that we have. This is the global statistics. We find the one with the highest value, and this is the one that the one gene SLC1787 that's probably most highly self-correlated inside of this data set that we have over here. And then we can plot those values over here when we additionally run like a local Moran's eye in order to get like local Moran's eye values. So areas that are showing brightly are areas where more transcripts are actually close together, or you have more counts that are close together. On top of that, we can also generate additional polygon information de novo. So this is really just here because we're really excited about the opportunity to just be able to analyze this information in like as many different ways as you want. So this is like how you might generate additional spatial units, essentially like additional sets of polygonal information you might want to use. So you can create, for example, like a hex bin grid to further aggregate your information this way, or you could also do something like a pseudo-Visium if you are interested in something like testing deconvolution results. And something that we're also working on recently is being able to do basically linked spatial querying. So that's what this is. If you were to basically clip one set of polygons with another. And just to mention again, the Giotto data package, this is where we have a set of mini Giotto objects, and there's objects that cover a wide range of different objects that are already pre-made, and they're pretty easy to load in and play around with. And Matt's going to talk a little bit more now about the other types of data that the other types of platforms that we're actually able to analyze.
Thanks, George. So as George just mentioned, the Giotto data package hosts a multitude of mini objects and sub-objects, which represent, for example, the expression data that we were looking at earlier, just so that people can quickly download, play around, get familiar with Giotto. And we support these for multiple different data sets. Something that's very important to the Giotto Suite team is that Giotto Suite remain technology agnostic. We have established tutorials and pipelines for the technologies that are on the screen on our website, which we will show at the end. It's giottosuite.com. You can access it now if you'd like. But today, I'm going to be taking us through a demo doing the Zenium human breast cancer, or with the Zenium human breast cancer dataset, excuse me. I'm just going to try to zoom in here. Was this good for everybody remotely and in the room? I hope that's visible. So to start out, we're simply going to load up our necessary libraries, standard workflow, and direct a variable to a location in which we can save all of our generated information. We can create a list of instructions that can be provided to the Giotto object just to specify how and where to save the information. From the Zenium experiment, we receive, pun intended, a suite of different files, including the H&E image, the immunofluorescence image, some metadata associated with the experiment, and subcellular files. Importantly, the cell boundaries, nucleus boundaries, transcript locations, feature metadata, as well as aggregate files for the expression and cell metadata. And generously, 10x also provides some cell typing files, which we will use at the end of this demo. For brevity, I'm going to skip over some of the metadata loading and explanation of the different probes for quality control. And we can get right into loading up the Giotto object. So the first thing we're going to do is load up our transcripts using data tables fread. We can filter that, we can filter that by Phred score and take a look at how many detections we are getting. And furthermore, as I said before, the expression information includes also transcript information includes some QC probes, including the blank code word, negative control, and negative control probe. So we can import those all into Giotto points after after we've just separated those out into a data table. Just a simple lapply, creating Giotto points, which is again a Terra spat vector under the hood, and we can preview those right away. I'm going to scroll past the QC. And we can take a look at specific genes right before we even put it into the object, just to ensure that everything looks the way that it should. Now we can import our segmentation data as George showed earlier. It's a rather simple pipeline. Read the segmentation data using data table fread. We can simply change up some naming conventions so it's readable by Giotto. Transform those from a data frame into Giotto polygons. And then we can plot those immediately just to make sure that all of the locations are as expected. Now we can create our Giotto object by providing a list of the different Giotto points that we've created representing our expression data, or transcript data, excuse me, as well as the polygon data in the form of our cells and our nucleus. Importantly, these keywords that we provide in the form of a list will form our feature types, and these keywords that we provide will form our spatial units. Now, that was a decent amount of pre-processing for one single experiment. So in order to evade this, we also provide a convenience function where you can direct create Giotto Zenium object right to a folder containing your Zenium data, and then specify if you want to do the subcellular analysis, aggregate analysis, and what boundaries you want to be looking at. Now, similarly to the previous demo, we need to generate an expression matrix from this. So we are going to take our polygon information and we are going to rasterize it and find the overlap with the transcript locations. We can view the spatial information that we've just created, seeing that we have right around 167,000 polygons. Now we can create our expression matrix calling overlap to matrix function. And as a sanity check, we can use the show Giotto expression function to verify that we do indeed have the same number of cells that are represented as we did polygons in our expression matrix, which is represented as features, or in this case, genes, by cells. Now, after this expression matrix has been created from the overlaps of the polygons and transcripts, it's rather standard to proceed along with normalization, filtering, dimension reduction, and clustering. But for brevity, we're going to skip over that as you know, a lot of packages have that capability, and that's not something we're necessarily looking to emphasize. Although, again, these scripts are available, so if you'd like to explore how we do that, we encourage you to. Now, earlier, I had mentioned that we can represent data by creating a spatial network, and it's rather simple just by specifying the amount of k neighbors we want and the maximum distance in a Delaunay network. We can create this network in one command and immediately visualize it afterwards. Although this looks a little convoluted, so we can get back to taking a closer look at this in a second. As mentioned previously, Giotto supports large image loading and viewing, which are, again, supported through Terra. We can plot these right away just to make sure that all looks good, and these are accessible throughout any given workflow with a Giotto object that is supporting images. So right there is the H&E, and over here is the IF, both of which are shown as provided from the 10x pipeline. So subsetting the Giotto object is also simple, but it's especially useful for our subcellular analyses when we can't really get a good picture of what's going on within the data at such a large scale when we have such fine resolution data. So we subsetted just to an arbitrary ROI, and we picked out 3 genes to plot along with the polygons just to take a look at you know, making sure that we got a good region of interest or something that you know is appropriate for this demo. So as you can see, the structures that were all created in the original object are preserved through subsetting, which is quite important for downstream analyses. One of which is the spatial network, which we can take a closer look at. Still rather busy at this resolution, but you can see nicely how these different cells are communicating with each other within that network. Now, an important part of supporting multiple spatial units is the fact that we can support custom segmentations. So in an event where you want to segment your own data and you want to see how those segmentations are performing, you are able to import those rather easily. So again, we use Terra under the hood, so I'm just going to import my custom segmentation in the form of a GeoJSON file. However, I've also done segmentations using CellPose, which exports mass files, which are usually like CSVs. Terra's vect function also handles that just as easily. This was created using Stardust, which is a QPath extension, and just right out of the box, we just wanted to take a look at how it did, and we decided to import it into our Giotto object. So it takes a little bit of manual manipulation because the segmentation was done on the immunofluorescence image, and we do not have the alignment data for the immunofluorescence to the transcript information. So after a little bit of manual manipulation and the same workflow as earlier, creating a Giotto polygon, adding that to the object, finding our centroids. This was hidden in the previous command. When we generated our polygons, we provide an argument for calculating the centroids automatically. We then find our expression matrix by calling these two functions, which we've shown already. And then we can run it through our pipeline, filtering and normalizing as necessary. And at this point, we wanted to take a look at a cell type enrichment to see just how different the overlap and generated expression matrices would be and what that would mean for downstream analyses in terms of cell typing. So using that list provided from 10x before, we performed a page enrichment, parametric gene set analysis enrichment on both sets of data, and we're able to look at histograms containing the distribution of different cell types. I do apologize, this text is rather small on this big screen, so I will zoom in for this portion. As we can see from this page enrichment for the original cell segmentation, the breast glandular cells are taking up the majority of this sample. So we can scroll down, and we run the same command just now for the Stardust spatial unit, which is our alternative set of polygons. And we can see a somewhat similar distribution, although it is a little bit different. One thing that we should be taking note of that I am going to emphasize now that I didn't previously is our frequency. So Stardust actually reported a lot fewer cells than the original segmentation did. Nonetheless, after these enrichments, we can take a look at the different kinds of distributions, comparing them directly, and we can see that our results change rather significantly. The amount of each cell type across both of our samples is different depending on our segmentation methods. And I'm not here to tell you that one segmentation is better than the other or superior to the other and that you should try alternative segmentation methods. What I'd like to emphasize here is that your segmentation is a key part of your downstream analysis, and ensuring a quality segmentation thus ensures a quality analysis. So we support the ability to host multiple segmentations within one object so that you can compare the two side by side and show your best possible results. Finally, we can save these Giotto objects quite simply by directing it to where we want to save it, specifying a folder name in which to save the RDS files. And this can be loaded in a later session by calling load Giotto and then specifying the path to which these, these RDS files exist.
Can I interrupt with an online question?
Sure.
So Ludwig Geislinger asks, says, it seems difficult to provide general infrastructure for the variety of spatial omics technologies while at the same time aiming at being technology agnostic. Which approach has Giotto taken to be technology agnostic? Does that mean you have specific functionality in your package that applies only to Visium as opposed to other functionality that applies only to Merfish?
So I can answer that a little bit. Yes.
On the mic. Thanks.
Yes, so I can answer that a little bit. The agnostic portion of it just means that we have all the tools needed in order to basically put together pretty much any set of spatial information that you have, regardless of the platform. So this can be subcellular information, it could be Visium information, and then we can actually analyze that downstream with aggregate methods. This does not mean that you can like actually do subcellular methods with non-subcellular data, not unless you want to do something like impute some data. That's going to be something else entirely. But we we have all of the tools, so that's why it's technology agnostic. We also do have some convenience functions that allow you to load in specific types of information, just like through predefined workflows that we have, mainly revolving around like, hey, we know specifically this is the output that we usually see from these packages or these platforms. These are the folder structures, and then after that, we can find like specific types of features, whether we want to filter those out for any particular reason, this kind of stuff.
Yeah, that's exactly right. I mean, I think the part about the convenience functions is really just streamlining the analysis and having to reduce the amount of, for lack of a better word, like data pre-processing that you need to do in order to actually import it into the object itself. Thank you for your question. So that, oh yeah, sure.
So actually I have a lot of questions.
Sure.
So we can talk more after the break. So one thing, okay, so as a background, I wrote spatial feature experiment in Voyager, which does something similar to what you're presenting now, but I presented last year. And when it comes to the transcript spots, like in some Murphish data sets, they're huge, like they're over 30 gigabytes. So how do you deal with the size of data, and how efficient is it? Like how much RAM does it take? Like how efficiently do you work with that?
Right. So that particular.
Sorry.
Right, so the package that you're
close?
talking about, or sorry,
Could you repeat the question
Yeah.
a little bit?
So the question was, how do we deal with scalability, right?
With the transcript spot.
Transcript
data
spot data.
Those can be like 30 gigabytes.
Yeah.
Yeah.
Specifically for Murphish, because that entails very large sizing up to around 30 gigabytes.
Yeah, we also had a lot of issues with that particular data set, and it was just very hard to be able to even put that in to memory. And you can't really deal with it even on servers because of just how big it is usually. But we are currently working on another package to deal with this kind of scalability in the form of like a database. So basically, we would transcode that into a DuckDB database, and then after that, we would be able to pull specific like spatial queries from there, and we would, that's our overall plan for how we would deal with this kind of thing.
All right, so that's my first question. So my second question is, do you have specific reasons why you use Terra for even the vector data instead of like SF? Also if I remember correctly from your first version of the paper for Giotto, you use a different data structure from single cell experiments. So is there a reason why you're not building upon single cell experiments? So you end up re-implementing a lot of the functionalities for like PCA UMAC and those.
I think just right off the tail end of your question. Yeah. That was a little bit of a long one. I think the summary of the question was, is there reasoning, and please correct me if I'm misstating, is there any reason for using Terra over something like SF to represent spatial data or transcript data, I believe you said in particular? And is there any reason why we wouldn't be building off of a single cell or spatial experiment and rebuilding these methods? So just right off the bat, you mentioned PCA. We do pipe in multiple bioconductor methods, especially for PCA being one of them, as an option for our users. But I think George can speak to a little bit more of why we didn't build off of spatial experiment or single cell experiment.
A lot of that was just because we already had the pre-existing infrastructure in the form of Giotto. So that's why we extended a lot from there. Yeah.
Do you think it would be like more interoperable with like an ecosystem of like existing tools like base, like also like a SpiceR, like there's a lot, like there's also Zelkenberg, like that community will make like AND data, so you can use like Python methods. So like do you think it's so do you think like not using single cell experiment would be like more of a limitation?
Sure. So the question was if not using single cell experiment would be a limitation, and if we have any considerations towards interoperability. One package that was brought up was AND data, and another that was brought up, was base. Thank you. So I actually wrote out an interoperability function to go from AND data to Giotto and vice versa. That is functional and live as of a couple of releases ago. We also have built out some interoperability functions for single cell experiment and spatial experiment, I believe. that was actually going to be addressed towards the back end of our presentation, but we are actively striving to be as interoperable as possible for that exact reason. We do not want to be limited by not being able to translate across packages. That's part of what motivates us, being a part of like the open source community, right? So I hope that answers your question.
So what about SF?
SF and Terra.
SF and Terra are newer generation ways of like dealing with this data, updates from like raster and SP, right? Terra is just like one of the newer ones that were on the field essentially, and we also found some pretty good benchmarks as to how fast it could process. So that's what we started with.
So related question, maybe a follow-up. Could you possibly highlight like what additional features the Giotto object has that the spatial experiment like?
Sure. So the question was, can we highlight specific features that the Giotto object has that spatial experiment does not?
So this new object that we are creating right now is able to handle things such as like transcript detections, so subcellular information, and also deal with polygonal information. So those things were not previously existing inside of spatial experiments specifically.
So can you say one more?
Yes. It provides functionality for dealing with subcellular data essentially. So it can work with points data in really efficient manners. It can also work with polygonal information as well. So those representations were not previously existing inside of spatial experiment.
One other thing I'd like to add, just in terms of methods, Giotto hosts a spot deconvolution method, spatial DWLS, which across several deconvolution reviews has been one of the most performant methods. So I think that is one notable feature about Giotto Suite as well. So we're going to continue along. So as we mentioned previously, as a result of these different spatial units and feature types, we are able to integrate multi-omic data. So this is especially notable as 10x has recently released the Visium cytosys co-expression dataset with both RNA and protein at the spot level. We are able to seamlessly integrate this information. And we also provide an interactive, Shiny application for manual annotation, which Joselyn is going to take us through.
Thank you, Matt.
You're welcome.
Okay, so for this section of the demonstration, I will be using a spatial site-seq dataset coming from Ronfan's lab. You can go to the paper and the original dataset following this link. So for the multiomics integration, we are using the WNN method that has been published before for the multiple individual modalities integration. Currently, we are able to support the integration of RNA plus protein modalities, but we are working on implementing the RNA plus epigenetic data, such as coming from ATAC-seq and also measurements from histones. The general pipeline behind the multiomics integration is to perform first the individual modalities preparation just in the regular way. So you can just select the different arguments for filtering, normalization, doing the the highly variable genes identification for the RNA, and the minimal step that you have to reach out for the multiomics is to calculate the PCA. The same thing for the protein modality. So you need to go to the filtering, normalization, etc. for the protein modality. I would just like to show you the results of the PCA and UMAP coming from the individual modalities. Sorry, the UMAP and the spatial. So this is the UMAP results together with the clustering for the RNA modality and recording them. And this is how it looks like for this data set, the spatial representation of these clusters calculated. For the protein modality, this is how it looks like, the UMAP resulting of this individual modality, and this is how it looks like, the spatial distribution of the clusters. Now for the multiomics demonstration, I will try to increases and go directly to the multiomics.
Yeah, we're still seeing the other window. So
I can adjust that.
cool.
Okay.
Sorry for infinity. It's not going anywhere. I might have to do a new share.
It should be fine.
Hi, everyone. Any luck on there?
The full window,
Yeah, I tried the window share. Yeah, it's cutting this off.
If RC has a pop-up, you won't.
Oh, just entire screen. Thanks. Still no luck. It's like cutting off the screen share, which is. Yeah, as soon as I go to share it, yeah, that was what I tried, and now this has a line through it.
So I guess let's just keep going. I gave them the link. They can see the HTML.
Okay.
So you could just go through the.
Yeah, let's go to the
in-person
HTML.
audience.
Yep.
Here in the,
Mm-hmm. Sorry.
Okay, so for the multiomics, basically you just provide the name of your previous individual modalities. In this case, it's the RNA and the protein, but information is stored together in the same Giotto object. So once that you get the results from the from calculating the W and N here, just while doing this calculation, you will see the progress of this calculation and the different steps that are within the W and N method. Once that you have these results that are automatically stored within the Giotto object, you can calculate again the UMAP, so that it will now take in count both modalities for each spot. And also you can recalculate the clustering, so that now each cluster will take in count both modalities. So this is how it looks like, the UMAP, just, considering the integrated RNA and protein data, and this is the integrated clustering. Now for the interactive selection, basically what you need to provide is a spatial plot that you can create also with Giotto using the spat plot function. And here you can either provide only the spots or the cells, depending on the level of your data set, and you can use or not the tissue image as reference. So here, ideally, we will just launch locally the Shiny app, and you will basically just plot here interactively the regions that you are interested on. In this case, I'm taking just as reference 2 different clusters that I previously calculated. But if you are more interested in the histological features, you can just remove the spots and leave the histological image. So once that you have selected these regions, you close the Shiny app that is just running within your R session. And the output for this function will be a table that contains the polygon IDs. You can establish the name that you prefer. And also you can zoom in or zoom out on this image in case you want to select very small regions on your tissue. So the output will contain the polygon IDs or their names that you provided for your regions, as well as the spatial coordinates. Now, this output lives outside of the Giotto object, so we need to integrate that information. Basically, just following these functions, you will integrate that information to the Giotto object, and as part of the downstream analysis from this interactive tool, you can just retrieve the cell IDs from each region that you have selected, as well as the non-selected regions, just as the background. You can also perform a differential expression between these selected regions, so you can get, for example, a heat map with some genes of interest for comparing your regions. You can also plot the abundance of your scripts, or from your transcripts or your genes of interest. You can perform also a comparison of the cell types abundance within each of these regions. In this case, I'm just making a comparison for each cluster, but if you have the cell type annotation within your cell metadata, you can plot just the comparison for each cell type. And you can also just make a comparison within the spatial plot for each of these regions in case you want just to analyze visually the abundance of each of the clusters or the cell types that you have in your data set. And finally, you can plot again your polygons just so that you don't have to do that again in case you just want to generate this figure of the regions that you have selected. And that's pretty much it regarding the interactive and multi-omics integration. I will pass to George for the final remarks.
Thank you, Joselyn. So back to here. So this is essentially our remarks about how we are currently working really hard in order for interoperability to be a reality with Giotto Suite and also the rest of Bioconductor. And we actually do have a script for that as well, but I'm not sure. We probably won't be running it in order to save time. We do have a quick demonstration here, just converting directly from.
Do you want to just zoom in a little bit?
Yeah, trying, but oh, there it is. Yeah, where we just start with a set of information that we load in from Giotto data, and then after that, convert it into a spatial experiment object. We perform NN SVG on it, and then convert it back into Giotto for actual plotting of it. And these are the results that come out of it. So that's just a quick demonstration of that functionality. And to finish up, there's really a lot that we're still not tapping into in terms of the R spatial ecosystem, which is like incredibly rich. And there's a lot of interconnected packages here that we could really use a lot of in order to further our analyses of biological information. So our future roadmap for Giotto Suite is that we're in the name, right, Giotto Suite. We're actually intending to turn this into a set of different packages that are a bit more modular and also separated in terms of their functionality. And we are also working on improving interoperability with other packages, both upstream and external methods. So for more tutorials, there's a lot more stuff that we haven't talked about. There's a lot of tutorials and examples that are available on the website, which is at giottosuite.com. This is a quick look at it. So we have installation, ecosystem, and we have quick instructions for things like how to create the object itself. The data sets are like entire run-throughs of different analyses that you have for different platforms. And we also have a section for tools that we're working on for detailing how you would do quick analyses, and specific things, specific actions in the Giotto object. So I think that's everything.
Just a quick comment on the website. I'm the newest, or one of the newest members in the Dries lab. So I've been working on this project for about a year. So if any of you are interested in using Giotto, my advice just to get started in the most streamlined way, definitely check out the Get Started section. And then I would look into tools and analyses before stepping into the data sets. The data sets are going to encompass workflows from both tools and analyses in a full-on analytical workflow. So going through data, getting started, tools and analyses first will orient you to then being able to use it at your own scale. Thank you very much for your time and attention, and we'd love to take any more questions if there's remaining time.
Yes. You were first.
I'm a bogan, I don't know if you know what
Interoperability.
Yeah.
It's a tough one.
It got brought up a few times.
Yes.
Right? Is there a plan to push this to CRAN or Bioconductor to kind of formalize it?
There is.
There is.
Style guidelines are one roadblock, among other things such as package sizing, all of which are are tasks that we can certainly complete. It is part of our plan. We would very much like to get on CRAN and or bioconductor. I think right now it's a little bit more of a bandwidth problem at the moment, though, or bandwidth obstacle, excuse me.
Automatic linting.
Yeah. There goes one of the problems.
So I have some questions related to how Giotto can be used for images only. I guess for the data size of the packages, experiment packages have a larger file size limit. That's a limitation for bioconductor. But going back to my question, though, for example, I think you mentioned cell pose, how you can incorporate segmentations from cell pose.
Yep.
So you could use, for example, an H&E staining image from Zenium, you can run cell pose, get where the cells are located. Then maybe you could have the Bayesian grid itself of the spots and their sizes. Can you use that to, for example, count? Can you use Giotto that way to count nuclei per spot? And then a related question is, for example, with RNA-scope, you can then run cell pose to also segment, find for the nuclei, or if you have a DAPI channel.
Sure.
But then at that point, your image has a different puncture, which is the location of your transcripts. So then could you then use Giotto there to count puncta per DAPI signal, or
Got it.
Or is it out of the scope of Giotto?
Sure. So I'll repeat the previous question. It was just about interoperability and getting this package on an open source mirror, such as CRAN and or bioconductor. I apologize for not repeating that earlier for the people that are virtual. The question right now is about utilization of an external segmentation method and integrating that with sort of this like idea of spatial locations, right? Merging your spot level resolution with, say, a Zenium experiment that is segmented, finding the nuclei, and kind of merging that together to find out how many nuclei and what information you can glean from one given spot. Yes, I believe so. And this was something that we touched on briefly, or George, excuse me, touched on briefly at the end of his VisGen mini data set tutorial with multiple spatial queries, where we had the hexbin tessellation. Let's pull it up. I apologize. We have this hexbin tessellation mixed with this pseudo Zenium analysis. So yes, in theory, this could be your nuclei that are within these Zenium spots. And then you could perform something such as, as I did in my walkthrough, some sort of cell type enrichment within each spot at the spatial unit level of your nuclei, for example, after importing that using Terra. So yes, that is possible.
I just want to ask, you have a bunch of things in Python and in R. So is the backend just, I'm curious how much of this can be isolated into one or the other. Let's say I don't have any, like I'm running completely on Andic data and scan Py. I want my pipeline to go into Giotto. Do I have any R, or intermediaries which I absolutely have to run Python for my workflow?
So is the question, would you want to stay in Python or would you want to stay in R? And is a bridge between the two required? I'm a little confused by the question. I apologize.
I'm trying to see how much of a dependency does this have.
Oh, how many dependencies does Giotto have?
Yes, because I'm going to import everything into one or the other.
Yep.
Requirements. How much do we have to regarding functionality before? Yes.
Do you want to take this one? The question was about Giotto's dependencies and leveraging or dealing with those in situations of interoperability.
Yeah, I think Giotto right now is around 30 different package dependencies for R stuff.
Yeah, so right around 30 mandatory installations,
Actually,
I believe. I don't have the number offhand. I apologize. But in terms of converting, I mean, it's really just a matter of what analysis you want to do. We've taken steps to reduce the amount of dependencies by removing certain packages from our import section and putting them in our suggests, as well as putting them in certain functions saying, if you want to run this kind of analysis, you need to download this package before running it. keeping these kinds of situations in mind. However, yeah, that would be an aspect of setting up before running any sort of interoperability command between the two.
So what you actually need, if you were trying to get this on the bioconductor or other areas, you would ask the community what part of the community you actually Python.
Sure. So the question was, if we were to try to make a big push to get this on bioconductor, what part of our package development would we ask for assistance with in getting this package onto one of those bigger repositories? I think George can better handle this question.
What part of
Our development.
our development?
Yeah.
Like, for example, do you need to use Basilisk for your Python code or not?
No, our Python code, we have we use our like Leiden clustering through Python, but the majority of the Python code that you're seeing on our repository is just the interoperability functions that I wrote, just to go from ingesting an AND data object, piping that into R using reticulate, and going back and forth. I'm not sure that I can best answer the question for how we can be best assisted.
If it's the question like which one do we have more packages of, it's
Yeah, we want to get this on. Yeah.
Maybe we could stop there and thank the speakers and you can come up and ask questions in person if you like.
Thank you all so much for your time and attention.